He wasn't down. His knee never touched. Oh my gosh, his knee never touched.
My name is Hannah, and I'm going to talk about the experiment that we did for the genetics lab BY210. So this experiment was designed to identify plant microbes in a specific Superfund site to see if we could identify any relations to these microbes protecting these plants against heavy metals. My Superfund site was the Alabama Plating Company in Vincent, Alabama, where 4 samples were taken from this site, and I specifically did samples 3 and 4. Multiple microbes were identified, and research was found to support protection against these heavy metals. A little introduction about the site. Electroplating happened at the Alabama Plating Company from 1956 to 1986. And this facility became a Superfund site in 2012 because of the contamination in the ground. And then cleanup happened shortly after plating ended, and so levels in the groundwater have been returned back to a safe level. So below are the methodology steps that we took to get this experiment going. So started with taking our samples, 1 sample from the stem, 1 sample from the root, and then we put them in test tubes, crushed them, and then we put a pH buffer, PBS buffer in there, and then we made petri dishes, and we used streak plating to grow a microbe culture on there. And then we continued along where we picked our best microbes, then we did genetic sequencing, and ultimately we were able to identify our plant microbes with these steps. So above are my results, and as you can see, I have sample 3, 1 lb and 100 lb, and then I have sample 4, 1 lb and 100 lb. And then here is a chart with some of the microbes that were identified. So I have Staphylococcus epidermis. The percent NCBI was 86.35, and this microbe uses cadmium to help grow biofilm for plant protection. And then I had Priestia megaterium. Percent was 97.20%, and this is used as a biocontrol, bioaccumulation, and it also helps with phosphorus solubilization and production of ammonia. Then I had Bacillus thuringiensis. This was 97.4%, and strain M318 helps as a protection. And then we had Pseudomonas rhizophyrae, and ZCR6 is isolated and used for bioremediation of sites co-contaminated with hydrocarbons and heavy metals. So the hypothesis that I came up with for this site is if plants are living in a contaminated soil area, there must be something that is helping protect it in order to help it thrive. So these microbes were discovered in this experiment, helped prove this hypothesis, because these microbes specifically have had a positive impact on plant growth. So these microbes are among the many microbes found to help plants thrive in a contaminated environment. They have either helped stimulate growth, or they have helped build the plant's resistance, or they have built some kind of protection around the plant to help them grow. And as a class, we discovered roughly 600 microbes that had a positive impact on plant growth. So here are my references that I used, and then I would like to acknowledge that this research was conducted by the BY210 Genetics Lab with the help of Banut Mohan, who was our TA, and Shahid Mukhtar, who was our